Hello and welcome to the first in our KDIGO webinar series, The Critical Role of Genetics in Kidney Disease, Not Just in Rare Kidney Diseases. My name is Bob Hanson and I'll be your moderator today. Some housekeeping notes. You'll be muted throughout the presentation, but please enter your questions in the box provided. We will cover as many questions as we can during the question and answer period at the end. Our hosts today are KDIGO, a nonprofit foundation developing international clinical practice guidelines in nephrology. In addition to its guideline work, KDIGO also holds 4 to 5 Controversies conferences every year, which bring together expert thought leaders to discuss and debate nephrology-related issues. In March of 2021, KDIGO will hold a Controversies conference on genetics and CKD, led by co-chairs Ali Gharavi and Anna Köttgen. KDIGO is honored to have their participation in this webinar today. KDIGO would also like to thank Natera, Inc. for their support of this webinar series. I'd like to introduce our speakers. Dr. Ali Gharavi, a leading kidney disease researcher, is chief of the Division of Nephrology at New York Presbyterian Columbia University Medical Center. Dr. Gharavi is an associate professor of medicine at Columbia University College of Physicians and Surgeons. Dr. Anna Köttgen, our second speaker, she is currently a full professor and director of the Institute of Genetic Epidemiology in Freiburg, Germany, and also an adjunct professor in the Department of Epidemiology of Johns Hopkins School of Public Health. Without further ado, I'll now turn this webinar over to our first speaker. Go ahead, Dr. Gharavi.
Hello, everyone. It's a pleasure to attend this webinar series on the genetics of CKD, and it's a pleasure to discuss our work on genomic testing for chronic kidney diseases. These are my disclosures. So I'm going to tell you about what we can do in terms of genetic testing for early diagnosis of kidney diseases. As you know, about 15% of the population in the United States has CKD. 10% of the population worldwide is estimated to have CKD. Nonetheless, the awareness of this disorder is fairly low in the population. Estimates in the United States is that globally about 10% of patients are aware if they have any level of kidney dysfunction, and even patients who have advanced stage 4 CKD, only about 60% of them are aware of it. And so this leads to an issue with late diagnosis, and often patients come in and are diagnosed with CKD of unknown cause because our standard clinical diagnostic tools are not sufficient to make a clear diagnosis. CKD is also prevalent in children. That's also another problem for diagnosing pediatric disorders very earlier on. The prevalence of CKD is increasing across the world, and so this issue is only going to get compounded over the years. The causes of kidney disease are different between children and adults. The most common causes of kidney failure in children are congenital and cystic disorders. About 50% of cases are attributable to these, and the remainder are primarily caused by primary or secondary glomerular disorders, and then various other entities. In adults, primary glomerular disorders and congenital disorders are still very frequent. However, as the population gets older, as this population in adults, then diabetic nephropathy and hypertensive nephropathy are more prevalent. So what can we do in terms of genomics to try to understand and get to the etiology of kidney diseases? The human genome sequence the first draft was published in 2001. There are 3 billion letters in the genomic alphabet. And so it took 10 years and $3 billion to generate that first draft. And subsequently, the real challenge was to produce the sequence of individual genomes. And so as the costs came down with the development of DNA sequencers and next generation sequencing, now this is quite feasible. Now it takes about 24 hours to sequence one human genome, and it costs about $1,000. So this is now another hundreds of thousands of genomes that have been sequenced nowadays, and we're able to compare these genomes to one another in patients who have specific disorders to see if there's a particular variant or set of variants that are predisposing to certain diseases. And that's really the challenge in front of us, is to look at these 3 billion nucleotides to identify the one typo that may be responsible for disease in a patient. I tell you about the genetic architecture of kidney disorders, and this is a classic graph depicting allele frequency of variants in the population versus its effect size. On the bottom right, we have common alleles that are fairly common in the population, and each one of them are typically associated with a low risk of disease. There are a number of genome-wide association studies that have been published over the years. You will hear from Anna Köttgen talking about her studies on hundreds of thousands of people to determine the variants that are associated with kidney function and related disorders. And we've done some studies on IgA nephropathy, identifying some genetic variants that are associated with disease. There's a lot you can do for these common alleles nowadays to be able to stratify patients with genetic risk, and we'll hear about it in the subsequent talk. My talk is really going to focus on the top left corner here, looking at really rare variants that result in kidney diseases. These rare variants are rare because they typically impart a large effect, and so as a result, they're purified out of the population. And so the rare variants are typically associated with these Mendelian disorders that we deal with, cystic diseases, tuber-interstitial disorders, congenital disorders. And altogether, all of these variants can also contribute to CKD of unknown cause, because this is one class of disease that can be, in which any of these disorders, congenital disorders, cystic disorders, FSGS can occur. And I'll tell you more about this. So what are our targets for sequencing? If we look at the genome, there are 3 billion nucleotides, but about 1% of it codes for genes. And so that coding portion of the genome is called the exome. And so there are no methods to just selectively sequence this coding portion, and that's called exome sequencing. If you look at the 20,000 genes and the exome itself, about 4,100 genes are associated with a Mendelian disorder, and that's sometimes called the Mendelium, that subset of genes associated with a monogenic disorder, and you can order a Mendelium panel for sequencing. If you look at that, again, a subset of the Mendelium genes, of those 4,100 genes, roughly about 625 are associated with a Mendelian form of kidney disease or genitourinary disorder. And sometimes this set of genes are called the nephrome. Within the nephrome, of course, there are, again, subcategories such as subset of genes associated with nephrotic syndrome or with congenital disorders. And again, you can order subpanels targeting these specific disorders. There are now efforts ongoing across the world to try to consolidate these gene lists and come up with a secure list of gene with disease associations. This is coordinated now by an effort called ClinGen, and they've done this for many other categories of diseases, and now recently they've turned to kidney diseases. And we hope that in the next few years, through this community effort, we will have a much better annotated list of kidney disease genes, which will facilitate the analysis and interpretation of genome sequence data. So the utility of genome sequencing became really clear as this technology was introduced, first as a research tool, but then the clinical utility was tested in a number of studies. On the left side, you will see two classic studies now looking first at about 250 cases that were mostly pediatric patients with neurodevelopmental disorders, and then subsequent follow-up with the largest series of 2,000 patients with of which, again, most of them were pediatric disorders, most of them were neurodevelopmental disorders, but you could see the diagnostic rate was about 25%. The other area where genomic testing has become quite routine is in cancer. On the top right, you see an example of a study of metastatic cancer, and these are mostly adults, or essentially all adults, and the diagnostic rate for germline mutations was 12.3%. We decided to look at this also in the context of chronic kidney disease and did a pilot study of 92 cases with early onset CKD, CKD of unknown cause, or CKD with very strong familial predisposition. And we identified a diagnostic rate of 24%, indicating that perhaps this is going to be a very promising approach for diagnosis and management of patients. We followed this up recently with a much larger study looking at 3,300 cases with chronic kidney disease. This was a cohort, two-thirds of the cohort was contributed from a biobank at Columbia, and then about 1,100 patients were contributed by AstraZeneca and the Aurora study, which was a clinical trial of statins in end-stage kidney failure. We wound up doing exome sequencing in this population, and Emily Groupman, a really talented PhD student who's now completed the thesis, did the variant annotation based on the American College of Medical Genetics criteria. These are clinical guidelines for sequence interpretation. The characteristics of the cohort, about 90% of them were adults. There was a good proportion of males and females, almost equal. We also had a multi-ethnic cohort. We wanted to make sure that we included a lot of non-European ancestry in our cohort because typically this category of patients had been omitted from many genetic studies. In addition, we also covered a large number of clinical diagnoses, including congenital disorders, glomerulopathies, diabetic and hypertensive nephropathy, but about 8% of cases also had CKD of unknown cause, which was, again, an important category for us to consider. This is the bottom line. Altogether, we had a 9% diagnostic yield for kidney disorders, and we had another 1.3% of cases who had another actionable variant. So altogether, we had 10% of cases, roughly, that had an actionable variant identified through this study. But the diagnostic yield really varied across different clinical categories. On the right, you see the diagnostic rate was about 1 to 2% for hypertensive and diabetic nephropathy, which are acquired diseases. And then if you look on the left side, we had the highest diagnostic yield for congenital and cystic disorders. The glomerulopathies had a diagnostic rate of 7%, so somewhere in between. The surprise to us was that CKD or ESRD of unknown cause had a 17% diagnostic rate, so roughly 1 in 5, 1 in 6 cases we could get a diagnosis in. And this was really promising to us, indicating that this is going to be a useful tool for clinical management, perhaps. I want to emphasize that the importance of making a clinical diagnosis after a nephrological workup for guiding our thinking about the diagnostic yield. These are patients who were seen by a nephrologist, evaluated, and then assigned to specific clinical categories. So somebody really thought about them and did a workup on them. I think the diagnostic yield for chronic kidney disease will be lower if simply patients are categorized based on an elevated creatinine and an EGFR. So I think the large population-based studies will tell us what is the overall diagnostic yield across the CKD population defined by epidemiological criteria versus looking based on a clinical workup by a nephrologist. Altogether, we had 66 diagnoses in 307 patients. About 2/3 of cases could be attributed to mutations in 6 genes, PKD1 and PKD2, diagnostic of polycystic kidney disease, the autosomal dominant form. And then we had a good large proportion attributable to mutations in type 4 collagen genes and also uromodulin. And the remaining cases were due to mutations in 60 distinct genes, so mostly occurred as ends of 1 and 2, really pointing to the great heterogeneity and the large fraction of really rare disorders seen in nephrology practice. The predictors of the diagnostic yield, besides the clinical categories I was telling you about earlier, included having a strong family history of kidney disease. And so this further emphasizes the need to ask for a family history during a clinical encounter. And then, as I mentioned, the different clinical diagnoses. Finally, we didn't have these data, but many studies have now shown that the early age of onset of kidney disease is also associated with a higher diagnostic yield. So I think this is another important factor to keep in mind. Young patients, early onset CKD would also be a good indication for doing genetic testing. Let me give you a couple of case studies. This was a 57-year-old with newly diagnosed kidney disease and blood protein in the urine. The kidney biopsy has shown focal sclerosis, and the patient was referred for steroid therapy. But he wanted a second opinion, and when we wound up looking at the workup, we realized that the patient, we weren't quite sure. There was some evidence of secondary FSGS on biopsy. So we wound up doing exome sequencing, and we found out that the patient actually has a mutation in type 4 collagen gene called 4A4, which was diagnostic of a dominant form of Alport syndrome. So the Alport syndrome, as you know, is a mutation that results in disruption of the glomerular basement membrane, and this is a structural issue. And it presents classically with hematuria, hearing loss, some visual defects, and some has pathognomic kidney biopsy findings, which we actually didn't see on this kidney biopsy. There are 3 genes that cause Alport syndrome. On chromosome X, the COL4A5, this is the classic form, X-linked form of Alport syndrome that affects, mostly males, whereas the autosomal form can have both dominant and recessive components attributable to mutations on COL4A3 and COL4A4. These mutations in COL4A3 and COL4A4 are increasingly recognized as a common contributor to kidney failure in the population. And so in this patient, we had to tell him that steroids would be of no help for him, and that we referred him for a new clinical trial designed for patients with Alport syndrome, and we started screening family members who may be at risk for the disease, and also trying to select individuals or family members who would be most suitable for kidney donation in case the patient progresses to kidney failure. So it turns out that these type 4 collagen mutations are the most common sort of underdiagnosed cause of kidney disease in the cohort we studied. We had altogether 92 cases, but only about 40% of them had a clinical diagnosis of Alport syndrome, a thin basement membrane disease. The others had carried a clinical diagnosis of congenital defects or FSGS or CKD of unknown cause. So this is something to really keep in mind as you're seeing patients with kidney disease, and they have a strong family history suggestive of a monogenic form of disease. This should come to mind. These type 4 collagen mutations should come to mind. And this also tells us that the classic presentation for type 4 collagen mutations, Alport syndrome, what we consider to be Alport syndrome, maybe not be the most common presentation for these disorders. So we have to keep these in mind. Here's another example, a case, a 25-year-old with a history of kidney disease since childhood. The kidney biopsies were done at age 8 and 11, but were non-diagnostic. And so he wanted to get married and wanted to get additional information about what might happen to his children. And so instead of doing yet another biopsy, we wanted doing exome sequencing. And here the diagnosis was based on the finding of a loss of function variant in CLCN5, which is diagnostic of a tubulopathy called Penn's disease. This results in low molecular weight proteinuria, hypercalciuria. There's, of course, bone loss, and nephrocalcinosis occurs due to the hypercalciuria, and kidney failure occurs in the patients in their 20s, 30s, and 40s. So there are actually treatments available for this with thiazide diuretics to reduce the hypercalciuria, a high citric diet, a number of other clinical trials available. Importantly, we made the diagnosis in 2 of his affected brothers, and he was told to go on a specific diet to start on the diuretics, and was also advised that because this is an X-linked form, his daughters would be obligate carriers, but then he would not pass this down to any family members, and so it's the next generation after that would have to be worried about it. Nonetheless, I think this was really important for their overall healthcare to make this diagnosis, not only to get the sort of the final diagnosis, but also because of the hypercalciuria, these patients are very prone to bone loss, and so he wind up going for bone density studies. And in fact, when we talked to the patient and to his brothers, even though they were young, vigorous men, they had already had multiple fractures in their medical history, which they attributed to just playing sports. So this diagnosis had important implications, not just for the kidney disease, but then for overall bone health and overall health in general. So this patient and this family basically had a CKD of unknown cause. This is a very important category, and so we recently reviewed 6 exome sequencing studies that have been published that included patients with CKD of unknown cause. Altogether, 443 patients have been published in the literature. Overall, there was a 22% diagnostic rate with exome sequencing. There were 47 distinct genetic diagnoses that were made, of which 29 or 62% represented singleton diagnoses, just really unique to that one patient. But our friends, the type 4 collagen genes, the mutations in those genes accounted for 1/3 of the cases. So based on the review of the literature, we came up with we proposed a workup for kidney disease of unknown cause. After and what we call kidney disease of unknown etiology really is after a thorough nephrological workup and standard testing has been done with or without a biopsy, if a nephrologist says, we really don't know why you have kidney failure, then you have to consider if there's a probability of a genetic diagnosis based on the family history, if there's consanguinity, if there's early onset of disease, particularly if there's cystic or congenital disorders. And then think about extra-renal manifestations, particularly other congenital defects. If that's the case, we recommend doing a chromosomal microarray and exome sequencing. Otherwise, if this is just isolated CKD without extra-renal manifestations, then maybe just do an exome sequencing, do a diagnostic interpretation. If it's positive, and then you can review the phenotype and this is all compatible, the diagnosis is compatible with the clinical presentation, then you can return the results. If you have still some doubts, you can review the results with a clinical geneticist or a nephrogeneticist to make sure that there's, why potentially there may be some discordance, or maybe there's a expansion of the phenotype that you were not aware of. If the results are negative, or you come up with a variant of unknown significance, but you still have a high suspicion of genetic diagnosis, you really have to review the data periodically, because there are new genes that are discovered over each year. And so as you review the data from year to year, the exome may be negative today, but it may be positive after you review the data a year from now, because a new gene for kidney disease has been identified. And if you think for some reason the exome sequencing has missed the diagnosis, then you have to consider additional tools to be able to interrogate the genome, such as whole genome sequencing, additional transcriptome sequencing, and there's a bunch of other analytic methods that can be applied to take this a little bit further, but it would require a specialized lab. But I think that, as you could see, just with plain exome sequencing, you should be able to get an answer in about 1 in 5 cases, at least based on the current literature. What are the genetic testing options that are better for patients? I'm often asked. So should we do a targeted sequencing of one gene? Should we do a gene panel? Should we do the whole exome? Or should we do the whole genome? For each of these approaches has their pros and cons. Targeted panels are optimized for the particular phenotype of interest. And so they have high coverage, high fidelity, sequencing. And so you should be pretty sure that you've interrogated those genes of interest at the time of sequencing. However, targeted panels need to be updated periodically as new genes are identified. And so if a targeted panel is negative and a new gene is identified, you can't go back and review the data. And that's where exome sequencing has an advantage, because now you're sequencing the entire, all the coding portions of the genes, nearly all of them. There are some gaps that you should be aware of in exome sequencing, such as some duplicated regions in PKD1. There are some genes that are just not captured. They'll cover some areas that are not covered by exome sequencing, and so that's something to be aware of. And then whole genome sequencing is a possibility. It gives you better analytic performance, but it's more expensive, and right now most of the data are not interpretable, the non-coding regions. So when we looked at those 3,300 patients who had exome sequencing, we tried to address this issue of diagnostic performance by creating virtual panels for cystic diseases, glomerulopathies, and tubulointerstitial disorders. And the way we did this is that we reconstructed these panels based on commercially available tests, and we used the union of these panels as a gene content. So we sort of stacked the deck in favor of panel testing. You could see that overall exome sequencing has a better performance, but it's pretty compatible when you look at the category of cystic or congenital disorders, glomerulopathies. But for CKD of unknown cause, because it encompasses all the different clinical categories of kidney disease, exome sequencing had the superior performance. So overall, I think that the panel testing is a reasonable first step for the sequencing for congenital disorders or glomerulopathies, but for other disorders it may not be as good. The other advantage of exome sequencing is that you get information about the rest of the genome. So there are a lot of mutations in other genes that may be actionable and diagnostic and important for your patient's health. An example of this is a 63-year-old who had a rare disease fibrillar nephropathy. We wound up doing sequencing, but really didn't identify any causes for her kidney disease. However, when we looked outside the genome at the list of actionable mutations, we found a mutation in BRCA2. At the time we tried to return the result to the patient had already had a mammogram that was diagnostic that found a breast nodule that had been biopsied and revealed to be breast cancer. So when we returned the BRCA mutation, though, this changed the management of the breast cancer because the BRCA2 positive tumor has to be treated very differently. So she wound up undergoing bilateral mastectomy, and then if there was advanced disease, there are specific drugs such as PARP inhibitors that have been shown to be much more effective for breast cancer, which when it's advanced, when we're doing cascade testing in family members to see if anybody else who might be carrier. In addition, in this patient, we had to think about what else did this finding do for us in terms of the management of the kidney condition. She was on immunosuppressive therapy, and we know that immunosuppressive therapy is a risk factor for future development of cancer. So essentially, we have to think about how to dose and time the immunosuppression in this patient, knowing that she has a BRCA2 mutation, and she may be prone to recurrence of tumors else, in other parts, in other organs in the body, in the uterus and in ovaries and so forth. So that's something for all of us to consider, because virtually every patient with chronic kidney disease either is getting immunosuppression because they have a glomerulopathy, for example, or because at some point they may undergo a kidney transplantation. And about 1 to 1.5% of the population has one of these actionable mutations that we would have to think about as we're trying to manage our patients. So overall, I can tell you that there's a lot of utility for genetic sequencing for kidney diseases for management. You can make a primary diagnosis of disease. Sometimes for the type of mutations can tell you about prognosis. For example, the loss of function mutations in type 4 collagen genes indicate a more rapid progression to kidney disease. There is, we can think about the risk of recurrence of kidney disease after transplantation. One can think about selection of therapy, such as avoidance of immunosuppression and output syndrome. There are risk factors that one can identify, such as APOL1 genotypes. We can screen family members to identify suitable donors for transplantation. There are additional information in the genome that can help you for thinking about surgical complications, such as the risk of thrombosis, cardiac events, anesthesia risk, and overall for the long-term health of the patients and thinking about comorbidities of CKD and disentangling the relationship of CKD to some of these comorbidities. One may be able to identify a risk of cancer, diabetes, and atherosclerosis, for example, by examination of the genome. And I didn't talk about pharmacogenomics, but that's also another area where probably there was going to be a lot of advances that will be made in the next few years as we try to identify genomic variants that are associated with variable response to drugs. So I want to thank a large number of people who participated in these studies in the renal genetics group at Columbia, all our nephrologists and pathologists who have been partners with this, our Center for Precision Medicine and Genomics, which aims to develop genomic testing across the board in internal medicine subspecialties, the Institute of Genomic Medicine, which funded the large sequencing study, and then the Columbia eMERGE cohort. Thank you very much for your attention. I also wanted to thank all of our funders for helping with these studies.
Okay, hello everyone. Thank you so much for your presentation, Ali. Dear colleagues, I'm very happy to be here today in this Genetics in CKD webinar series and present some findings on the current landscape, what we currently know about the landscape of kidney function in health and disease. I have no disclosures. As Ali already said, CKD has a very high prevalence of more than 10% in the adult population, but despite the high prevalence, we currently treat it mostly symptomatically. The reason we treat it symptomatically is that we still know relatively little about the etiology, or that the etiologies are very heterogeneous, and we hope that we can use the genetic component of kidney disease to gain mechanistic insights into these etiologies and also to use them for prediction. Nowadays, we can use genome-wide, often referred to as unbiased, modern techniques to sequence the genome and then carry out these genome-wide screens with the advantage that we can gain novel and unexpected insights by not focusing on a particular candidate gene of interest. Thanks to technical advances, we can now screen hundreds, thousands of individuals at single base pair resolution, making these genome-wide studies very powerful. So in my talk, as opposed to the previous talk, I'm going to focus on these common variants of small effects and what we can do with knowledge from such studies and why we believe they're important. As I mentioned earlier, if we deal with small effect sizes, then consequently, in order to detect them, we need to study very large populations. So we need to study many thousands and hundred thousands of people as opposed to a couple of hundred of patients. This is typically done in population-based studies simply because population-based data are readily available in large sample sizes and because CKD has high prevalence. So even in a general population-based sample, there's going to be a large fraction of people with kidney dysfunction of some degree. And our main objectives in population-based genetic research are A, to quantify the contribution of genetics to kidney function. Second, to uncover molecules and pathways that can provide some mechanistic insights into kidney function and disease and may represent a basis to develop more specific therapies. And third, to predict the risk for disease initiation and progression and to improve diagnostics. And this third point is also possible if we don't understand anything about the molecular pathways, even simply by having a good predictor. So as I said earlier, population-based research is often carried out in large studies, and these are not large enough by themselves typically, so these studies typically form consortia. And one worldwide consortium in which we study kidney function genetics is the CKD-GEN or CKD-GEN Genetics Consortium that I co-direct with my colleague Christian Patero from Bolzano. And in this consortium, which is an international collaboration of approximately 125 studies, we combine data from more than 1 million individuals, where on each person we have information on genotypes of 10 to 20 million genetic variants per person. So we can scan the genome very precisely. We have an analysis group. This is a group of researchers who does the hands-on work and the heavy lifting. We meet once a week in a virtual lab, and then we have a common server and computing infrastructure where we develop the analysis plans. We provide scripts that are dispatched to the studies. We quality control the data, and we combine them through statistical techniques such as meta-analysis. And we also do bioinformatic characterization of findings. So I thought that for each of these objectives, I'm going to give one or two examples to make this a little bit more concrete. The first objective is to quantify the contribution of genetics, because obviously if we want to use genetics to learn something about etiology or to predict disease course better, then of course we're looking for phenotypes or traits that have a large contribution of genetics. In our most recent publication from the CKD-GEN Consortium, which appeared in 2019, we combined data on genetic variants and their association with estimated glomerular filtration rate, or EGFR, as the most commonly used measure of kidney function. And combining data from all these individuals resulted in the identification of 264 genetic regions where we found genetic variants that were reproducibly associated with inter-individual differences in kidney function. You can use this data to estimate the heritability of EGFR, meaning how much of the variation in the inter-individual variability in EGFR can be attributed to additive genetic effects. And doing so for estimated glomerular filtration rate resulted in an estimate of 39%. If we now quantify quantify the proportion explained by these 264 loci identified, it's about 20% of the estimated heritability, or in other words, 7% overall. And you can say 7% is not a lot, but and there's a lot of missing heritability, so there's a lot of unexplained heritability. But what you see in this graph is that over time, as genome-wide association studies grew larger, we could explain more and more of the genetic contribution to kidney function. So even though we're studying very large samples, increasing the sample size further explains more of the heritability. So as a summary for the first point, currently we can explain about 20% of the estimated contribution of genetics to inter-individual differences in EGFR. The second objective is to uncover molecules and pathways that may provide mechanistic insights into kidney function and disease. And here I'm just going to show one example. The example comes from a publication that came out last year where we combined data on genetic genetic associations with albuminuria, or the urinary albumin to creatinine ratio, as a measure of kidney damage. The data was combined across different ancestry groups from more than 550,000 individuals and resulted in the identification of 68 loci, which you see here just labeled with the closest gene on the outside. And I don't want to focus on the details of the plot, but I want to make the point that among the genes that were identified, these are all common genetic variants, there were many biologically really interesting and plausible candidates. So for example, we found common variants in cubulin, which encodes for the receptor that's responsible for albumin reabsorption from the urine. We identified variants in one of the collagen genes, and we identified genetic variants in this gene, which encodes for the mineralocorticoid receptor. This is interesting because the mainstay of treatment to lower UACR is inhibition of the renin-angiotensin-aldosterone system, so of the very system for which this gene encodes the receptor for aldosterone. So this shows that although the effect sizes are small, these studies can identify genes that are suitable targets to modulate the phenotype, as exemplified here. Switching back to Ali's slide, you may remember that collagen 4A4 was one of the genes he focused on as causing a rare genetic disease. And so the point we take away from this is that monogenic versus complex genetic studies are really complementary, and they can implicate the same gene. So if collagen 4A4 is important in the function of the kidney, there may be some mutations that are very detrimental, have a large effect size, and cause monogenic disease. And there may be some variants that are more regulatory, maybe just tweak the level of collagen 4A4, and hence can rise to higher allele frequencies. And so it's really important to view these studies as complementary and not contrasting and in separate domains. Okay, switching back to my example, I want to focus on one particular challenge that we have in these population-based studies, and that is not the case in clinical exome sequencing. And that is the challenge to really find the causal gene and elucidate the mechanism by which it causes kidney disease. And I'll show you what I mean in the next slide. Fortunately, there are novel tools nowadays, novel computational tools that we can use to address these challenges. So what do I mean by this? This is zooming in on a genetic association we identified. So on the x-axis here you have the chromosomal position, on the y-axis you have the negative log 10 p-value of association, and each of these dots is one genetic variant. So you would say, oh, here on chromosome 11 there is a genetic variant with this name, and it's associated with a measure of kidney damage with a p-value of 10 times, of 10 to the negative 10, so very small. And you can also see that there are several genes in this region, as illustrated here, so we don't really know which is the causal one, and especially that this genetic variant maps in between genes. And this is what I mean by that it's more difficult to know which is the relevant gene than in clinical exome sequencing studies, where we typically have truncating or missense mutations that affect the exome, so the coding part of the gene. So what can we do to get closer to which is actually the relevant gene in this region? Well, we can assume that if these associated SNPs are in between genes, then probably their role is that they regulate the expression of one of these genes. And if they do, then they should also be associated with either the expression levels of the gene that is causal, but not of the other genes, or with an encoded protein. And we can test this. And without going into detail, there's statistical methods to test this. And if you just focus on this shape, what the association between the genetic variants and kidney damage looks like, you can immediately appreciate that we get the same shape when we look at these same genetic variants and the expression levels of this OAF gene, which is this gene here. And this matching shape was not detected for any of the other expression levels of the other genes, only with this one. And likewise, when we look at plasma protein concentrations of the proteins encoded by all of these genes, we only find a matching shape when we look at the concentrations of this OAF gene. And so what we can deduce from this is there's a genetic variant. It leads to lower expression of this gene in kidney tissue, so we can also look at different tissues. It leads to lower protein levels and then to higher albuminuria. And if we know this, then we can formulate a mechanistic hypothesis and say, well, if lower expression of the gene causes higher amounts of albumin in the urine, then this means that either the glomerular filter is damaged and more albumin gets into the urine, or the albumin reuptake in the tubules is reduced. And so we cannot as efficiently reabsorb the filtered albumin. There are a number of model systems, obviously, with which you can now study which is the case, because obviously we would like to know, is the filter leaky or is the reabsorption impaired? And of course, mammals are great, but the generation time is really long, and we want to get a clue a little bit quicker. So one example, one model organism you can look at are Drosophila. These are fruit flies. And they don't have kidneys, but they have cells, nephrocytes, that perform the function of the kidneys, which is they remove material from the circulation through a combination of filtration. So they also have SLIT diaphragms, followed by endocytosis of albumin here in these slits. And so these cells reflect both the filtration and the reuptake. And we can study a fluorescently labeled tracer protein and see if one of these functions is impaired. Without going into experimental details, what you can do is that you can, if we suspect that this OAF gene is responsible, we can suppress it and then study if the tracer molecule behaves differently. And so if the control flies over here, you can then see that reducing the abundance of this gene, knocking it down, resulted in significantly reduced reuptake. This is another positive control gene, and this is OAF, resulted in significantly reduced uptake of the marker, which supports that the human ortholog, so the human version of this protein, has a role in either glomerular filtration or tubular albumin reuptake. And to distinguish this, you can then use imaging studies where you can look at the surface of these cells. And I don't want to go into this in detail, but what we found is that these two rows here correspond to OAF, that because the glomerular filter looks as under control conditions, it's likely that the damage that we observe is really due to reduced albumin reabsorption in the proximal tubule. So the point I wanted to make is that genetic studies followed, they can give us information about what experimental follow-up to perform and which cells to look at and how to go forward. And this is much more efficient than having to test every single gene in one of these regions. And this is all done computationally prior to the lab. Okay, now I'm going to shift gears completely and go away from the mechanistic insights because obviously these take very long. If you remember, we know the causal genes for polycystic kidney disease for over 20 years now, and we still don't really understand on a molecular level what happens in ADPKD. So while it's interesting to give a basis for molecular studies, we cannot expect that mechanistic insights will occur very quickly. However, we can use these information, these findings from these common variant studies clinically to predict risk for disease initiation and progression, and also to improve diagnosis. And I just brought 2 or 3 examples to make this point. One example is based on a study of IgA nephropathy out of the Columbia group, where Ali Gharavi and Christoph Kirilluk looked at more than 7,000 IgA cases and 12,000 controls of East Asian and European ancestry, and identified 15 such genetic loci where there were genetic risk variants associated with IgA nephropathy risk. What you can then do is, based on these 15 independent genetic regions in the genome, you can pick out the variant with the strongest effect at each of these regions and sum them up. So sum up how many of the risk alleles are carried across these 15 variants by a given person. And because a person has 2 chromosomes of each type, this score ranges from 0 to 30 if you have 15 risk variants. And you can also weight this by the effect size of the variants. And then, because the score is not distributed equally but normally in the population, you can group neighboring categories and look at quintiles, for example, to get more robust estimates, which is what was done here. So on the x-axis, you can see the quintile of genetic risk score. So these are the people in the lowest 20% of genetic risk, and these are people in the highest 20% of genetic risk. And on the y-axis, you see the mean age at diagnosis of IgA nephropathy. And so what you can appreciate is that the higher your genetic risk of IgA nephropathy was, the earlier the diagnosis was made, which is presumably related or a proxy of severity of disease. Another example from IgA nephropathy that I think is interesting is a study by the same group where they constructed a genetic risk score based on 7 SNPs. This was an earlier study, in fact, and then compared the distribution of this genetic risk score across different ancestral populations. And they found that African Americans on average carried fewer high-risk alleles for IgA nephropathy than did Europeans, here in yellow. And the highest genetic risk was found in individuals of Asian ancestry, as shown here in red. And this matches very closely our clinical observations that IgA nephropathy prevalence and incidence is far higher in Asia than in Europe and higher there as compared to Africa. You can think about why this is the case, and the most likely explanation that we currently have is that as people have migrated out of Africa, something in these genetic risk variants for IgA nephropathy provided a selective advantage with respect to other things. So, for example, response to bacterial infections. And this is why these risk variants for IgA nephropathy rose to higher frequencies and are more commonly found in these populations. And this is a common theme for genetic risk variants that have relatively large effects, yet are common. It's also found in APOL1 in sleeping sickness, and we also observe this for membranous nephropathy, even if we don't know yet what the selection pressure is. For membranous nephropathy, this works likewise. So this is a study that recently appeared in Nature Communications by Chris Kirillok and team, where they looked at 3,700 cases and 9,000 controls, identified 7 genetic regions on which they built a risk score, and then used an external sample to look at how the risk score was correlated with clinical characteristics. And what they found is that this time the risk score was grouped into deciles, so each dot is 10%, so these are the people who are genetically, the 10% of people genetically at lowest risk, and these are the ones at highest risk. And here on the y-axis they looked at the odds ratio, so the risk of being affected by the disease. And you'll find that this has a approximately 30-fold gradient depending on your genetic makeup, which is quite impressive. They also found significant differences in the proportion of people who were positive for phospholipase A2 receptor antibodies, and as the risk score increased, also the proportion with albuminuria was higher based on 24-hour urines. I mentioned that you can not only use genetics to predict risk better, but you can also use genetic risk scores to improve the diagnosis of diseases, which was also done in the paper I just introduced. And what you can see here is that here the investigators combined a genetic risk score termed GRS with a combined risk score termed CRS, which combined phospholipase A2 antibodies, receptor antibodies, and the genetic risk score. And if you look at the area under the curve, which should take this shape here for perfect specificity and sensitivity, you can see that adding genetic information makes this a much better test, as illustrated by the much higher area under the curve. So genetic information on top of clinical antibody tests really helps to diagnose membranous nephropathy. And this could also be compared to people without kidney disease, to people with other kidney diseases, so IgA nephropathy or FSGS, and illustrated that this genetic risk score is really specific for membranous nephropathy. So the distribution here the properties of this combined test were really specific to membranous nephropathy. Lastly, you can use these genetic risk scores not only to predict the disease itself, but you can also use it to predict consequences of a kidney phenotype. So, for example, when you look at serum urate, this is some of our own work, the blood concentrations are very much controlled by the renal excretion of urate. And we performed a genetic study where we identified 114 genomic regions that were associated with differences in serum urate. We constructed a risk score based on these and then used it to try to predict gout, which is a consequence of hyperuricemia, in an independent sample. So this was completely independent. And we found that across categories of the genetic risk score for urate, the prevalence of gout, or here you can see the odds ratio of gout, you can, and so the chance to be affected by gout varied 100-fold. So these genetic risk scores are really quite powerful, and interestingly, 3.5% of the entire population that we studied have a genetic risk score that conveys a risk of gout that is comparable to one of the monogenic diseases that cause gout, but obviously much more prevalent. So in summary, I hope that I could show you that population-based studies of kidney function and disease can provide estimates of the contribution of genetics. They can provide a foundation for mechanistic studies, and they can deliver genetic scores that we can use for risk prediction and diagnostics. Meta-analyses of genome-wide association studies have sufficient sample sizes to reliably detect associations between common genetic variants that convey small effects. But small effects do not mean that the implicated gene is not important. It just means that this genetic variant doesn't have a very dramatic impact. But a full loss of function mutation, as shown by collagen 4A4, can still have a very large impact on the function of the gene. Monogenic and polygenic studies can identify the same genes, which are just important in kidney function in the healthy range, but also for causing kidney disease. And classical challenges for genome-wide association studies can be addressed with new data integration methods to identify causal genes and tissues. I would like to close by thanking my colleagues in the CKD-GEN consortium, a huge number of investigators, study personnel, and participants, too numerous to name them all, the KDIGO team for the help with preparing the presentation and for the opportunity to present, my team here, my local team, and our funders. Thank you for your attention.
Thanks very much. Yes, we do have some questions. Reminder to type your questions in the box provided, and we'll get to as many questions as we can. Our first question is, in the future, will it be more useful to do research based on GWAS and population studies, or WES in CKD patients to learn about the genetics of kidney disease?
Yes, thank you. That's an important question. I hope I could illustrate that it really depends on your objective. As Dr. Gharavi showed, whole exome sequencing in CKD patients is really the way to go when you need to provide a diagnosis for a patient presenting with kidney disease who may have a family history and has a clear suspected monogenic kidney disease or kidney disease of unknown cause. Genome-wide association studies are complementary in that they may identify genes for which simply no patient has been seen for a monogenic disease yet, so they can yield novel findings. They are better at getting or they provide complementary information in getting at pathways because different people have different variants, in different genes. And you can use them for risk prediction, which is something that requires larger samples. So I think it will both be useful and really depends on your objective.
Okay, thank you. Our next question is, what is the optimal approach for current clinical application? In your opinion, exome, large comprehensive panel, or smaller targeted panels?
So this is a very important question that comes up often. I must say that we don't have any studies that do a direct head-to-head comparison of all of these different modalities. As I mentioned, we did a sort of a virtual comparison looking at our exome data sets. I think that in general, the diagnostic yield really depends on the clinical category of disease and your clinical certainty about or suspicion about specific disorders. If you're pretty secure about sort of what kind of phenotype you're dealing with, you're sure this is nephrotic syndrome, and this is you should look at the gene panel for nephrotic syndrome, and you will have a diagnostic yield. That's going to be still smaller than exome sequencing based on our study, but still it's going to be pretty good. If you're not entirely sure about the phenotype or the diagnosis, then exome sequencing is probably the way to go, and then you can always go back to exome sequencing even if you don't get an answer, to be able to reinterrogate the data as you have new genes that are discovered. So my approach would be to look at the phenotype, think about your degree of certainty about the clinical diagnosis, and from there decide whether you want to do a phenotypic driven panel versus an exome sequencing. If you're not entirely sure if you have the clinical classification right.
Okay, next, what are some examples of personalized medicine? For example, tailored drug dose, choice based on disease classification, etc.
So I think right now the best, the most common application, I would say, for exome sequencing or genetic testing is in nephrotic syndrome, when clinicians want to understand whether they should pursue immunosuppression or not in somebody who has nephrotic syndrome. And so here it's more about avoidance of a drug and avoiding side effects. We know immunosuppression is associated with long-term side effects. And so if you find a patient who has nephrotic syndrome due to mutations in, let's say, a podocyte gene, then you can really spare them a lot of complications of immunosuppression, and particular steroid therapy using that way. If you think about the targeted therapies, one has to be clear about it. There are enough sufficient examples in nephrology. Examples that one can think about it is, for example, for Fabry disease. If you make that diagnosis, then there's enzyme therapy that's available. There are mutations in the CoQ10 pathway that result in nephrotic syndrome, and then sort of he gives CoQ10 supplementation, then you can mitigate against those findings. So I think that there are a few good examples, but not a sufficient number of them. The hope is that as we identify more genetic disorders, we are able to study these a little bit more precisely and come up with targeted therapies much better. And I would also say that perhaps outside of cancer, there aren't that many good examples of targeted therapy anywhere in clinical medicine. So we should not feel bad that we don't have any of those yet in nephrology, but we hope that in the future we will have many more.
Okay, thank you very much for that. Next question is, the effect sizes detected through GWAS are so small. How can this be of any clinical relevance?
Yes, so this is a question that I get often, in fact, and I can relate that sometimes it's disappointing when reading these large research papers to see how small the effect sizes really are. But I hope I could show that in the case of collagen 4A4, as an example, this gene can be identified through whole exome sequencing of patients presenting with monogenic manifestation, or through genome-wide association studies of a milder phenotype, just the amount of albumin in the urine. And this, so this shows that the common variants detected in population studies may have a small effect, but if you have a complete loss of function of this gene, the effect can be very dramatic. And the same is true, for example, for the uromodulin gene, UMOD. And an example that's often cited is from the field of hypercholesterolemia, where common variants in the gene encoding for HMG-CoA reductase, which is the target of statins, they have a very small effect on LDL cholesterol levels. Yet, if you inhibit them pharmacologically, then you can lower LDL cholesterol by 60%. So just because something has a small effect size in a GWAS doesn't mean that it's not a good target.
Okay, on behalf of our audience, I want to thank our speakers for their presentations today. Questions that we did not have time for will be forwarded to our speakers for individual follow-up. Watch your email for more information on the next webinar in our KDIGO webinar series. At this time, we will end the webinar and you may now disconnect.